replication occurs in both eukaryotic and prokaryotic organisms. DNA has a double helix structure composed of several nucleotides held together by phosphodiester bonds between the phosphate and 5-carbon sugar and hydrogen bonds between the nitrogenous bases. A nucleotide is composed of a 5-carbon sugar, a phosphate group, and one of the 4 nitrogenous bases: adenine, guanine, thymine, or cytosine. Adenine and guanine are purines, while thymine and cytosine are pyrimidines. There's also a 5th nitrogenous base called uracil, which is a pyrimidine, but it is only in RNA. The bases are specific when they are paired with the 2 backbones. A pyrimidine only pairs with a purine. More specifically, adenine pairs with thymine and guanine pairs with cytosine. The 2 strands of DNA run anti-parallel, meaning that the 5' to 3' direction of one strand runs counter to the 5' to 3' end of the other strand. A sugar is considered to be 5' if the phosphate group is attached to the 5-carbon of that sugar. If the phosphate is bonded to the 3-carbon, then it is a 3' end. Replication begins at the origin of replication, where the helicase separates the 2 backbones. The unwinding creates a bubble and a replication fork. Then single-stranded binding proteins hold the 2 strands apart, and this is where DNA replication begins.
Of the new strands created during DNA synthesis, one of them is the leading strand while the other is the lagging strand. They are named as such because the leading strand is much easier to synthesize because it only needs one group of RNA primers added to the molecule to allow the synthesis of DNA to be possible. Primates will add the 2 molecules to the new strand, one at a time, and the new molecules will latch onto each other at the 3' end of the molecule. Only one group of primates is needed because DNA polymerase III runs in the direction of the 3' end of one molecule to the 5' end of the next, because the 5' is already occupied by a phosphate group. DNA polymerase III will attach to the newly created strand, adding one molecule at a time. Creating phosphodiester bonds between the original molecules that were already in place from the primers and adding on, building the new strand continuously. Then, DNA polymerase I will come and remove the existing primers that were added and replace them with the correct DNA molecules, one at a time. Creating phosphodiester bonds between the newly created molecules.
The lagging strand is a much more complex process than the leading strand discussed previously. The lagging strand is much more complicated because synthesis must always go towards the replication fork, but DNA polymerase III can only run in the direction opposite to where the strand is traveling. For this process to occur, it starts off the same. Primates will come and add in primers that will be used as a basis for the strand to complete replication, one at a time, adding once again to the 3' end of the molecule. However, multiple primers are added, unlike the leading strand, so that DNA polymerase III will have a reference point of where to stop. Primates will add one molecule and then another molecule to the 3' end of the previous molecule. Now DNA polymerase III will come in again and add new molecules in between the new gaps, again running from the 3' direction to the 5' end of the other molecule. Then DNA polymerase III will realize that it has hit a stopping point as it has created phosphodiester bonds between these molecules. Once it is finished creating this bond, it will move back to the beginning and run until it hits another RNA base that has already been added. It will add a molecule and then another molecule to the 3' position of the previous molecule, leaving space open not binding to the RNA that was here previously. After this process has been completed, creating Okazaki fragments, DNA polymerase I will once again come in and remove the already established RNA bases and replace them with the correct DNA fragments, once again creating a bond between the newly added fragments. It does the same thing with the other bases. And then finally, an enzyme called ligase comes and attaches Okazaki fragments to the recently replaced RNA primers, creating a phosphodiester bond where they were attached.